As we send people on long-term missions farther and farther into space on missions beyond low Earth orbit and the Moon, we need to know more about the physiological risks in order to solve the considerable obstacles to human space exploration. Physiological problems such as weightlessness, immune system changes, or bone loss during long-duration human exploration can be better understood in the microgravity environment aboard the ISS. These are studied and explored together with the crew and the biological model systems. Space biologists are also studying the role of Earth's organisms, animals, plants, and microbes in long-term life support systems and biohabitats in space or on planetary surfaces.
Space biology is NASA's hypothesis-driven, discovery-based biological science in space and the associated technology that goes along with that. Human health is a very important part of space life sciences, and space biology is this discovery-based animal and plant and cell research that leads to those new countermeasures for going beyond low Earth orbit. In space, cells do change, and we want to understand not just what's changing, but how the proteins that are changing are working in the cell and how that's adapting and responding to the space environment.
There are several microscopes on board the ISS to aid space biologists in their investigations. The Light Microscopy Module, or LMM, has already helped physicists and engineers develop novel advanced materials and improve consumer products, both for Earth and in space applications. The LMM is ready to provide a unique vantage point from which to study the effects of space on biology. Using a modified commercial Leica RXA laboratory microscope, the NASA Glenn Research Center has configured the LMM to allow real-time interaction and manipulation of experiments in space and ground-based controls. Interaction with the experiments is now routine, based on experience for staff, researchers, and the in-flight ISS crew. The LMM is located inside the Fluids Integrated Rack of the U.S. Destiny Laboratory module aboard the International Space Station. The FIR rack provides the LMM with common laboratory infrastructure, including an optics bench, temperature control, power, illumination, imaging, data processing, and isolation from vibrations so that high magnification images can be captured without jitter. The Light Microscopy Module uses the existing electronics of the Leica microscope, supplemented with unique and innovative internal and external electronics to support remote operation, enhanced automation, and superior imaging. Motors and linear actuators motorize the microscope's manual functions. Engineers at NASA Glenn Research Center equip the Leica with 23 micromotors, which permit remote control by scientists on the ground and reduce ISS crew time requirements in space. This tends to increase the available time for research. Two identical high-resolution black and white cameras can be mounted on the headpiece, one coaxially in the viewing axis of the microscope. The other can be mounted at an angle in the tube assembly. Additionally, a small surveillance camera is available to be mounted inside the auxiliary fluids container, where sample cells are processed and contained. Imaging techniques include bright field and fluorescence from 2.5 to 100x. The Flight LMM microscope fluorescence turret has 8 locations which are occupied with the following filters: FITC, Texas Red, a polarizer, half mirror, DAPI, a 50-50 beam splitter cube, red, blue, and green. Plans are in development to reconfigure the filter turret by replacing the red, green, and blue filters with a TRITC filter cube, mirror, and a blank location for confocal use. The capabilities of the red, blue, and green pseudo-color can be replaced with a color camera when required. The LMM has previously imaged regular and specialty microscope slides, including Petri dishes and OptiCells, which allow oxygen transport through a membrane and provide for containment of the biological sample and the sample medium.
The benefits for being able to view the experiment as it develops on orbit is huge, because being able to see things as they are functioning in the environment in which you're testing, because by the time you get the plants back from orbit, they've changed. And so if you can't capture those images immediately or very soon while they're being challenged in the environment of the space station, you lose those critical data. And so you really have to be able to view them in situ.
The LMM can support 5 main areas of space biology with an orbit viewing of materials before gelling or freezing: plant research, cell culture research, microorganism research, invertebrate research, vertebrate research, macromolecular biophysics.
I am a NASA biomedical engineer and lead developer of our VestGen software. This fractal-based software maps and quantifies vascular remodeling for human, vertebrate, and even invertebrate model systems. We're also associating the geometric, dynamic dimensions of vascular remodeling and blood flow with essentially unlimited numbers of bioinformatic dimensions. These informatic dimensions of localized protein and other molecular expression are captured by fluorescence microscopy that is available to you now on the LMM for your space biology experiments, with confocal capabilities planned in the near future. These approaches are illustrated in our VestGen mapping analysis of vascular tissues in ISS model organisms that include the mouse GI and retina, fruit fly wings, vascular patterning in Arabidopsis leaves, and even the human retina for our research on astronaut ocular and vision problems. We invite you to picture your space biology research using the LMM and potentially other microscopes manifesting in future ISS missions until 2024.
Currently, the microscope offers backlighting and epifluorescent illumination, high-resolution color video microscopy, dark field, and phase contrast capabilities are also being developed. A commercial condenser has been proposed to provide bright field illumination. The FIR and LMM upgrades for a confocal scanner, a neodymium YAG laser, and faster digital cameras are manifested for 2016. This will enable 3D structures to be analyzed in more detail.
Using the LMM for biology is a bit of a challenge. It's not designed for biology per se, and so a biological scientist, has to adapt their biology to be able to fit both operationally and physically into the unit. However, being able to view things in situ at this kind of level of resolution at the microscopic level, also with fluorescence, is a huge advantage to much of the kind of biology that many of us do, and it's certainly worth the effort.
Space biology experiments scheduled to be flown and observed in the LMM over the next several months include APEX 3, or the Advanced Plant Experiments on Orbit, flying on SPX F5 and 6. Generally, there are 30-day docking periods that are available for space biology experiments before the physics colloid experiments begin.
The LMM can aid space biologists, physicists, chemists, engineers, and graduate student researchers in achieving a better understanding of biological samples, colloids, and other physical processes on the microscopic level. If you're planning a project that could benefit from in-flight observations in the Light Microscopy Module, there are several ways to proceed. You can use the CASES program, a NASA research announcement, or contact your program executive at NASA headquarters to get on the flight manifest.